Today, I'll be talking about TAAC reaction, Topochemical Azide-Alkyne Cycloaddition reaction, and its uses. So before I do that, I will shortly and briefly introduce my research. I do research on other aspects also. For example, I do natural product synthesis, not very complex molecules, simple molecules, and also we are interested in other aspects of supramolecular chemistry than topochemical reactions. Today, I will be talking only on topochemical reaction. Let us see what we can do with topochemical reaction. So there is a lot of interest in getting crystalline polymers. Usually, when you make polymers in solution, you get an amorphous material. And as you know, crystallinity has a lot of property. Crystallinity offers property to materials. So there is a lot of interest in crystalline polymers. One possibility to make crystalline polymers is by topochemical polymerization. Topochemical polymerization means the polymerization in the crystal, actually. So we make monomers in such a way that they arrange in a fashion such that they can undergo easy reaction. For example, in our monomers, there are two different motifs. They are complementary to each other. For example, this red head and this blue head are complementary to each other. That means when they are in proximity, they will react and form a new bond. So now we design this kind of monomers in such a way that they will undergo this kind of self-assembly in the crystal, and they make this kind of chains, and whereby they make this blue head and red head at proximity between monomers in the crystal lattice. And if so, we are bringing these two these reactive motifs at proximity, then the crystal will undergo reaction upon mild activation by heating, and we'll get a polymer, which is crystalline actually, because our monomer is crystal, so the polymer has to be crystalline. So we get crystalline polymers in this way. This is a topochemical polymerization. Most of the time we get crystals, but in very rare occasion we get amorphous material also, because after the reaction the crystallinity will be lost, but that's very, very rare. Now, this is the reaction we were interested in. This is azide-alkene cycloaddition reaction. If you heat this one, this is called Huisgen reaction. This will undergo cycloaddition, 1,3-dipolar cycloaddition, to give these kind of two isomers. Actually, this is triazole, 1,4 disubstituted triazole and 1,5 disubstituted triazole. You get a mixture of material if you just heat these two reactants in solution, for example. But you know, the copper catalyzed click chemistry, that will give only this isomer. But we cannot use any catalyst or solvent in the crystal state. We have to use only the crystal. So why we are so interested in this particular cycloaddition reaction is that this is highly exothermic reaction. This is the energy profile of that reaction, coordinate of that reaction we can see. So this is azide-alkene energy somewhere here, and the product is somewhere here. So there is a lot of energy reduction, or it's an exothermic reaction, about 65 kilocalorie per mole. And this is the peak of the hill or transition state, you can say, wherein this will start forming a bond and this will also start forming a bond. So in this state, these two are in parallel orientation. Now, it is possible that we can make this kind of orientation or close to this kind of orientation in the crystal lattice. That means this reaction will undergo automatically without any catalyst or any other mode of activation. So let us see how this we can do this one. So this is the first example we have done. This is a galactose derivative having azide and alkane, complementary reactive motifs. And this is a fresh crystal of this material, and this is a 40 days old material. Crystallinity is similar, actually, morphology is same. So you think that this is also the same crystal, but it is not so actually. It is a mixture of several oligomers. If you see the TLC, this is a mixture of all kinds of oligomers. And if you isolate some of these material, you can see all of them have undergone oligomerization. We could isolate some of these oligomers by column chromatography. And also we can do mass spec. This is multi-spectrum. You can see different oligomers equally spaced because each monomer is getting added here. And this particular material is undergoing reaction at room temperature, even at 0 degrees centigrade, without doing anything. Just make the monomer crystal, it will undergo to polymer. If you if you do the crystal structure, this is the crystal structure alignment. This is the azide here and alkane here, and they are proximally placed about 3.3 angstrom unit between them. And this is the packing arrangement. They are packed in a head-to-tail fashion. That means if they react, you will get a linear polymer actually. So after some time, if you redo the structure, after collecting the data again, after some time redo the structure, what we see is completely it's reacted, and you have a polymer which is linked by this kind of triazole. So all this azide and alkane reacted to form a one-dimensional polymer. And this happens at room temperature, even at 0 degree centigrade, actually. So it's very facile reaction. Just time brings the reaction, nothing else. No activation is required in this case. So can we design such topochemical reactions, more topochemical reactions? Then we looked at the Cambridge crystallographic database actually. So all these structures are reported actually, crystal structure, and all of them have this peculiar diol motif. And because of this diol motif, all of them undergo this kind of zigzag hydrogen bonding. Now this is what we want to use, and we want to make this kind of monomer having azide and alkane having this important diol motif. And this is what is expected based on the CCDC data. And you can say it's a supramolecular fibril. It's a hydrogen bonded fibril. And if you look along the fibril, this is what you see. So the fibril is going behind the screen. And in the crystal, of course, you know that there will be several such fibrils. And this is the most probable arrangement possible in the crystal lattice because of a possible azide-alkene attractive interaction. There is a dipole interaction possible, also pi interaction possible. If so, I am placing this azide and alkene at proximity, and they can undergo easy reaction to give this kind of glycopolymers wherein these two sugar units or many sugar units are connected by this kind of triazole linkage. To see that, yes, we have made this material simple chemistry. I will not bore you with the synthetic scheme. So essentially, we made this material having azide and alkane and this important structural motif. And this is crystallized. You can see the crystal picture. And this is the packing diagram. The zigzag hydrogen bonding you can see as expected it is there. And in the other direction, this is the AB plane of the crystal. In the C direction, you can see this azide and alkane are at proximally placed in a chain-like fashion. So in a head-to-tail fashion. So they can undergo reaction in principle. So now upon heating this material, the crystal is intact, no melting happens. So after heating about at 90 degrees centigrade for about 24 hours and redo the structure, what you see, the hydrogen bonding is intact. Only difference is all the azide and alkane vanished, and in that place you have triazole linkage actually. So this is the polymer, glycopolymer, which is formed by this monomer, just simple heating. So this is what we make a crystal with the crystal of the monomer in such a way that this azide and alkane are at proximity. Then we heat slightly and we get a crystal. Of course, we start with the crystal and we get the same crystal, but it's a polymer now, monomer to polymer. This is what we do in this kind of chemistry. So now an important chemistry we have done is here. So this is a disaccharide called chachalose, 2 glucose units connected through this anomeric linkage. Usually this cannot undergo further polymerization or polymerization because both the electrophilic centers are connected through this bond. So you don't have electrophilic center anymore, or you don't have anomeric carbon free. So we thought we can make polymer using our chemistry. What our plan was to make this azide at this hydroxyl position and alkane at this hydroxyl position. Then can we make polymer out of this one? So again, simple chemistry, we made this material and we got 2 crystals, two polymorphs of the crystal from 2 different solvents. This is ethyl acetate and hexane, and this is chloroform and hexane, 2 different polymorphs. And both of them have very similar packing, except that in the chloroform crystal, you have chloroform molecule also. And there, of course, you can say solvatomorphs. I mean, they have in one of the form, you have solvent actually. So you can see the powder hexadi. And both of them, if you heat this material, both of them undergo reaction in a crystal to crystal fashion. And yeah, this is how you monitor the reaction using NMR actually. So at different times you withdraw some amount of material and dissolve it in some solvent, whichever solvent it is soluble in, and monitor what percentage is reacted. This is about 96% reacted sorry, 93% reacted in 96 hours, and similar reactivity for the other foam also. Both foam reacts to give the polymer, Trahalo's polymer. And next, of course, the next hydrogen bonded system is nucleic acid, actually. So when again we went to the CCDC database, all these structures are reported. All of them are cytosine derivatives. All of them have this particular arrangement, actually. This is an NHN dimer. Initially, this cytosine derivatives form a dimer, and also they form hydrogen bond along this direction through NHO hydrogen bond. It's a tape, actually, molecular tape in all these crystals. So this is very concept. Now, this is what we want to use to design DNAs, actually. So if we have a cytosine derivative, this C for cytosine, and 5' and 3' with azide-alkene, this is what the arrangement we expect in the crystal because of the cytosine dimerization. And by this way, we are placing the azide-alkene at proximity, then we can do reaction along this direction, and we can make a DNA or DNA analog wherein you don't have a phosphodiester linkage, but you have a much more robust triazole linkage. Let's see how we do that. So this is what we wanted to make. So we have made, again, simple chemistry, I mean, tedious chemistry, but non-chemistry, actually, not very complex chemistry. So we made this material. As expected, this is forming hydrogen bonding dimer, and also it's forming tape, molecular tape along this direction. And by this way, this azide and alkane are at proximity, and this undergo reaction. If you heat, this is the starting material NMR, and this is the product NMR. It just completely reacted up to 10 mer we can see in the MALDI spectrum, because higher mers are insoluble in water, actually. So this is, again, crystal to crystal. We have monitored this reaction by different powder oxide and other techniques, and it's a crystal to crystal reaction. Now, this is a simple, I mean, another derivative of that, you have an extra benzoid here at this end position. This N-benzoyl cytosine derivative, and that has an additional pi-pi stacking, because of which the distance between the azide and alkane are much more closer, and this is ready to react, actually. So just by heating at 60 degrees centigrade, this undergo cycloaddition reaction, you get polymer. This is the polymer, or the DNA, the structure of the DNA, homobasic DNA structure we got through this single crystal to single crystal transformation. So now, next system, of course, is protein. So protein is also highly, I mean, amino acids are highly hydrogen bonded system. Can we make proteins, actually, pseudo proteins? So this is our strategy here. So small peptides, which are not from beta sheet, they undergo beta sheet, and by that way, they will place their head and tail between adjacent stacks or adjacent sheets at proximity. This is anti-parallel beta sheet, actually. So you can see this head is this direction, and this head is in this direction. This is anti-parallel. This is the parallel one here. All heads are on the same direction. In either case, between adjacent stack, one stack, another stack, between adjacent stack, the head and tail are at proximity. Even in here, when it is anti-parallel, they are at proximity. Now we wanted to make these kind of peptides, small peptides, wherein our head and tail are azide and alkane. We can do reactions. So this is what we want to make, and this is what is expected. And we'll be placing this kind of azide and alkane at proximity if you make this kind of small peptides. And then we can do reaction along this direction. We can make pseudo proteins or larger protein-like molecule. So the first sequence we have taken is valine alanine system having azide and alkane, crystalline material. And this is the hydrogen bonding direction or beta sheet direction. You can see the azide and alkane between adjacent stacks are at proximity. And this undergo reaction. Upon heating, it undergoes reaction. Again, by NMR, we have monitored the reaction. About 90% reacted in 200 hours at 85 degrees centigrade. So also, you can monitor by IR spectroscopy. You can see the vanishing of this azide signal slowly due to the consumption of this azide in the reaction, cycloaddition reaction. So in this case, we could not get the final crystal structure, but still it is single crystal to single crystal. You can see before heating, after heating, this is the optical polarizing microscopy image. You can see the birefringence. Birefringence means this is crystalline. So there is no amorphous character, but we could not get the crystal structure of this, and I will tell you why in a moment. So nevertheless, we got very high polymer. I mean, up to 7 kilodalton. I mean, the size of a small protein, actually. And this also shows, this is the powder oxide, time-dependent powder oxide is showing it is a crystal to crystal transformation, which is happening. And it's a complete about in 130 hours, it goes to completion. So you get a protein-like molecule. So now why we could not get the crystal structure is here, actually. So
So this is a fresh crystal, and this is an aged crystal. After 1 month, we see all the crystals are twisted in the RB flask, all of them in the right-handed helical fashion. So we have NMR'd this one, and we found that about 10% reaction happened even at room temperature. Now we have taken a fresh crystal and we heated it. And in few minutes, you can see this is twisting in, again, right-handed helical fashion. Now, these are made up of amino acid, which is chiral, actually. So now the question is, why always a right-handed helical fashion? Does the chirality have any say in the direction of twisting? So naturally, we made the enantiomer of this material, that is the DD peptide. Initially, we made LL peptide. This is the mirror image of that. And in that case, we found that the twisting is in the opposite direction. This is, you can see, this is a crystal which is twisted many times. This is another crystal, of course, of opposite enantiomer, and that is twisted in another direction. So this is the first paper which is showing that this kind of molecular chirality has a say in this kind of microscopic response by crystals. So also, fortunately, this is a good gelator and makes gels with water and organic solvents, and the gel fiber also undergo reaction in this way, I mean, in a topochemical way, because gel is also crystalline, but it's a different crystal. This is the crystal powder hexadide. This is the gel powder hexadide. They are different. That means they are different polymorphs. And this polymorph, that means the gel polymorph, also undergo reaction. And we can get some kind of size control since it is a gel. So this also we have reported in gel state. Of course, another system which we wanted to take is this kind of diphenylalanine derivatives. This is known to, this kind of diphenylalanine known to form a beta sheet assembly in various papers. Actually, it's already reported. What we have added is just an azide-alkene. This is new. So we expect this kind of arrangement, and we can do polymerization along this direction. So we made this material, and this is the packing diagram. You can see the azide and alkanes are trapped in this kind of cavity, and they can undergo reaction. And at 60 degrees centigrade, this can undergo complete reaction. This is monomer, this is polymer. And in that process, and this is a single crystal to single crystal transformation, after polymerization, you can redo the crystal structure, and you can see that it's completely reacted and formed polymer along this direction, like this. And you see two dots. These dots are water, actually. The water was not there in the monomer, but during the reaction, there is space created because of slight motion in the crystal, and this takes 2 molecules of water per monomer unit from atmosphere contemporaneously with the reaction. That means as the reaction goes, this will take water from atmosphere. And there is a lot of interest in such material which can take water molecules from atmosphere. Usually people are interested in such atmospheric water harvesting. And usually they use MOFs, metal organic frameworks. The problem there is metal can bind with water very strongly, and it cannot release water so easily. And also, after a few cycles of hydration, dehydration, we don't know whether the MOF will have its structural integrity. That's a problem. But this is a simple organic material, no metal. This can take 2 molecules of water. That means 8.1% by its weight it can take. And this is reversible. It can take. We can see we have done 30 cycles of hydration, dehydration. Every time we can do the crystal structure. This is the anhydrous form. This is the hydrated form. We can do even single crystal XRD. And the interesting thing is that this water, this is showing the TGA, which is leaving. This water is leaving at very low temperature, less than 50 degrees centigrade. Even at room temperature, this water can be released. So that's a good thing about this particular organic material, which takes water, unlike MOFs. And also, at what relative humidity it can take water is very interesting. This can take water from 30% relative humidity. That means in Saudi Arabia, for example, with desert countries, it can take water. So it's very powerful to take water molecules. And this is very much reversible. And there is a lot of, as I said, there is a lot of interest in such molecules which can take water. All these things are single crystal to single crystal process. Monomer to polymer and hydration to dehydration is a single crystal to single crystal process. Now, another aspect I want to tell you is packing dictates property. We know these are different forms of carbon, diamond, graphite, fullerene, of course, other forms of carbon also. And all of them have different properties. And these properties are dictated by their packing. Now,
can we make different packing of the same polymer and can get different properties? Now, this particular material, we got 3 different polymers from different states actually, and all of them are different actually. You can see from the powder XRD spectrum, they are different polymorphs, and all of them have different packing actually. These are packed in an anti-parallel fashion, the monomer, and this is parallel, this is also parallel, but there is a difference between these two because there are 2 different kinds of molecules here. Here are only one, conformers. Here only one kind of conformers. So all this will give different polymers if it's reacted. All of them are in a ready to react orientation in the crystal. So we have heated all of them, and in all the cases we got polymer in very good yield, and all of them have different properties. As we did not study all the properties yet, we are still studying. So for example, they have different mechanical property, mechanical strength, and for example, solubility. Other studies are going on actually. So this is what. So we make a monomer, and we make different polymers of the monomer, and we do the TAAC chemistry or TAAC polymerization to get different crystal forms of the same polymer, and all of them have different properties. So this is the same spectrum, same this one. So another one is glycine. I think I have few more minutes I have. So this is glycine, polyglycine. There is a lot of interest. This is a glycine repeats actually. This is present in some of the natural proteins like silk, spider silk, for example, or even other silks. And silk gets this strength because of this particular domain. And it has 2 different forms, polyglycine 1, polyglycine 2. This is a normal lamellar packing. That means each chain is connected to 2 other chains. And this is a hexagonal packing. That means each chain is connected to 6 other chains. So this is called polyglycine 2. So we wanted to see what will be the structure or packing of this kind of polyglycine, wherein one amide unit is replaced by this kind of isosteric replacement, that is a triazole. So we wanted to see every 3rd amino amide group is replaced by this kind of triazole, what happens, or every 4th one, what happens. So we naturally made 2 different monomers, this monomer to make every 3rd one triazole, and this one to make every 4th one as a triazole. So both of them crystallized in a very similar fashion. Actually, this is the crystal structure of this material. You can see the azide and alkene in a ready-to-react orientation. And this is not beta sheet actually. This is crisscrossed actually. They are 90 degree. Adjacent layers are at 90 degree to each other. So this undergoes reaction at room temperature as in the first case. So after a few days, it completely reacts to a polymer actually. This is the polymer. And since we have a crisscross monomer, what you get is cross-laminated polymer. This is a new topology for polymers. Actually, this is not synthesized of this kind of topology. Polymers in this kind of topology. So this is the next one, the next higher actually. This is the tripeptide. Again, very similar packing, very similar distance for reaction. So they undergo reaction without anything. At room temperature, slowly undergo, or the monomer evolved into a polymer slowly at room temperature. And in this case, so you get a cross-laminated polymer. And this is the cross-lamination. Even common man knows cross-laminate is much more stable than this lamellar packing. So this is the cross-laminate structure we got. These are the structures already known for 1D polymers. This is normal lamella. This is the interwoven structures, all are reported by Omariagi and co-workers. In all these interwoven structures, you have metal ion. Of course, for interweaving, you need metal ions. This is the cross-lamination we have reported. There is no metal ion. That's a good thing. And of course, we have shown that this cross-laminated polymers are much more stable, thermally stable than the random polymer we made from the same material. This is the random polymer decomposition temperature. And this is the temperature at which the crystalline polymer decomposes. So there is a property because of this particular packing actually. So I think a few more minutes I have, yes. So this is another one, 2 different polymers in a single crystal. Can you think of that? Can we make 2 different polymers in a single crystal? So this particular sugar derivative, we crystallized and we found that they have 2 different, there are 2 different forms actually, conformers, and they are self-sorted in such a way that this line will form only 1 5-substituted triazole. And the next line will form 1 5-substitute 4 substitute triazole. So this will give one kind of polymer, this will give different kind of polymer, although the monomers are same, but the linkage is different. So this is what we get, a mixture of, and 1:1 mixture of two different polymers in the crystal. And this is a perfect blend, you can say. This kind of blended structure also we have made in a single crystal fashion. This is a new topology reported for amyloid structures. Usually amyloids are beta, cross beta. So beta sheets stack one over the other and get these amyloid structures. A cross alpha amyloid was recently reported by Lando in 2017 in a small peptide obtained from Staphylococcus aureus, a bacterial peptide. So this is a new topology for protein scientists, for example. So this is, so can we make this kind of topology? So exactly, we have made this kind of topology wherein we have this kind of helical peptides, which are again stacked through hydrogen bonding. And this is just similar to this kind of cross alpha amyloid structures. And they have better properties. I will not have time to discuss about those. And this is another topology we have designed. Again, this leucine isolation system are known to form this kind of P65 packing. Actually, that means every adjacent peptide will be at 60 degrees to the previous one. So at 360 degrees, you will have 6 molecules in a different angle, actually. So if you polymerize this kind of thing, we will be having a twist stacked by every layer is stacked, twisted by 60 degrees, twist stack topology for the polymer. So this is what we have shown in this latest paper, actually, a twist stack topology in the polymer. And yeah, I think because of time, I think I should stop here. So I have briefly discussed about what is a topochemical azide-alkyne cycloaddition reaction, and we have shown this can be applied to make polymers of different sugars and sugar-like molecules. And also we can take the same strategy to nucleic acid to make DNA or DNA mimics. We have many more results in this area, which you will see in future soon. I mean, then we went to peptides. We can make peptides and make pseudo proteins, and we have made again several pseudo proteins. And we also do other topochemistry. This is alkyne PDA or diacetylene polymerization. We do topochemistry on this also to make different functional polymers. Now, I should thank my hardworking students for all these results. These are all the graduates from the group. This is Hema. She is also currently graduate, and she is in Weizmann Institute now in Israel. This is the current group, and I thank all this funding agency, ISA Toronto, and finally thank you all for your time and also for the opportunity.